everyone, I am Shwelidash welcoming you to the Medical and Pharmacy Crash Course Division in the Knowledge Club online. At previous episode, we learned about the function of nervous tissue. Today, I will discuss on the action potential. By the end of this section, you will be able to describe the components of the membrane that establish the resting membrane potential, describe the changes that occur to the membrane that result in the action potential. Stay tuned up to end because if you skip the lecture, you can miss a valuable points in between lecture for your exam. So stay tuned up to end. I am going to start today's episode. Let's start. The functions of the nervous system, sensation, integration and response depend on the functions of the nerves underlying these pathways. To understand how neurons are able to communicate, it is necessary to describe the role of an excitable membrane in generating these signals. The basis of this communication is the action potential, which demonstrates how changes in the membrane that constitute a signal. Looking at the way these signals work in more variable circumstances involves a look at graded potentials. Electrically active cell membranes. Most cells in the body make use of the charged particles ions to build up a charge across the cell membrane. Previously, this was shown to be a part of how muscle cells work. For skeletal muscles to contract based on excitation-contraction coupling requires input from a neuron. Both of the cells make use of the cell membrane to regulate ion movement between the extracellular fluid and systole. As you learn this chapter on the cells, the cell membrane is primarily responsible for regulating what can cross the membrane and what stays on only one side. The cell membrane is a phospholipid bilayer, so only substances that can pass directly through the hydrophobic core can diffuse through unaided. Charged particles, which are hydrophilic by definition, cannot pass through the cell membrane without assistance. Transmembrane proteins, specifically channel proteins, make this possible. Several channels as well as specialized energy-dependent ion pumps are necessary to generate a transmembrane potential, and to generate an action potential of special interest is the carrier protein referred to as the sodium-potassium pump that moves sodium ions out of a cell and potassium ions into a cell, thus regulating ion concentrations on both sides of the cell membrane. The sodium or potassium pump requires energy in the form of adenosine triphosphate, means ATP, so it is also referred to as an ATPase. As was explained in the cell chapter, the concentration of sodium ion is higher outside the cell than inside, and the concentration of potassium is higher inside the cell is higher than outside. That means that this pump is moving the ion against the concentration gradient for sodium and potassium, which is why it requires energy. In fact, the pump basically maintains those concentration gradients. Ion channels are pores that allow specific charged particles to cross the membrane in response to an existing concentration gradient. Proteins are capable of spanning the cell membrane, including its hydrophobic core, and can interact with the charge of ions because of the varied properties of amino acids found within specific domains or regions of the protein channel. Hydrophobic amino acids are found in the domain that are opposed to the hydrocarbon tails of the phospholipids. Hydrophilic amino acids are exposed to the fluid environments of the extracellular fluid and systole. Additionally, the ions will interact with the hydrophilic amino acids, which will be selective for the charge of the ion. Channels for cations, the positive ions, will have negatively charged side chains in the pore. Channels for the anions, the negative ions, will have positively charged side chains in the pore. This is called electrochemical exclusion, meaning that the channel pore is charge specific. Ions can also be specified by the diameter of the pore. The distance between the amino acids will be specific for the diameter of the ion when it dissociates from the water molecules surrounding it. Because of the surrounding water molecules, larger pores are not ideal for smaller ions because the water molecules will interact by hydrogen bonds more readily than the amino acid side chains. This is called size exclusion. Some ion channels are selective for charge but not necessarily for size, and thus are called a non-specific channel. These non-specific channels allow cations, particularly sodium, potassium and calcium, to cross the membrane but exclude anions. Ion channels do not freely allow ions to diffuse across the membrane. They are opened by certain events, meaning the channels are gated. So, another way that channels can be categorized is on the basis of how they are gated. Although these classes of ion channels are found primarily in cells of nervous or muscular tissue, they also can be found in cells of epithelial and connective tissues. A ligand-gated channel opens because a signaling molecule, a ligand, binds to the extracellular region of the channel. This type of channel is also known as an inotropic receptor because when the ligand, known as a neurotransmitter in the nervous system, binds to the protein, ions cross the membrane, changing its charge. A mechanically gated channel opens because of a physical distortion of the cell membrane. Many channels associated with the sense of touch, the somatosensation, are mechanically gated. For example, and pressure is applied to the skin, these channels open and allow ions to enter the cell. Similar to this type of channel would be the channel that opens on the basis of temperature changes, as in testing the water in the shower. The voltage-gated channel is a channel that responds to changes in the electrical properties of the membrane in which it is embedded. Normally, the inner portion of the membrane is at a negative voltage. When that voltage becomes less negative, the channel begins to allow ions to cross the membrane. A leakage channel is randomly gated, meaning that it opens and closes at random. Hence, the reference to leaking. There is no actual event that opens the channel. Instead, it has an intrinsic rate of switching between the open and closed states. Leakage channels contribute to the resting transmembrane voltage of the excitable membrane. The membrane potential, the electrical state of the cell membrane, can have several variations. These are all variations in the membrane potential. A potential is a distribution of charge across the cell membrane, measured in millivolts, that is MV. The standard is to compare the inside of the cell relative to the outside. So, the membrane potential is a value representing the charge on the intracellular side of the membrane based on the outside being 0. Relatively speaking, the concentration of ions in extracellular and intercellular fluids is largely balanced with a net neutral charge. However, a slight difference in charge occurs right at the membrane surface, both internally and externally. It is the difference in this very limited region that has all the power in neurons and muscle cells to generate electrical signals, including action potentials. Before these electrical signals can be described, the resting state of the membrane must be explained. When the cell is at rest and the ion channels are closed, except for leakage channels which randomly open, ions are distributed across the membrane in a very predictable way. The concentration of sodium outside the cell is 10 times greater than the concentration inside. Also, the concentration of potassium inside the cell is greater than outside. The cytosol contains a high concentration of anions in the form of phosphatidylate and negatively charged proteins. Large anions are a component of the inner cell membrane, including specialized phospholipids and proteins associated with the inner leaflet of the membrane. Leaflet is a term used for one side of the lipid bilayer membrane. The negative charge is localized in the large anions, with the ions distributed across the membrane at these concentrations. The difference in charge is measured at -70 MV. The value described as the resting membrane potential. The exact value measured for the resting membrane potential varies between cells, but -70 MV is most commonly used as this value. This voltage would actually be much lower except for the contributions of some important proteins in the membrane. Leakage channels allow sodium ion to slowly move into the cell or potassium to slowly move out, and the sodium-potassium pump restores them. This may appear to be a waste of energy, but it has a role in maintaining the membrane potential. Now come to the point, the action potential. Resting membrane potential describes the steady state of the cell, which is a dynamic process that is balanced by an ion leakage and ion pumping. Without any outside influence, it will not change. To get an electrical signal started, the membrane potential has to change. This starts with a channel opening for sodium in the membrane. Because the concentration of sodium ion is higher outside the cell than inside the cell by a factor of 10. Ions will rush into the cell that are driven largely by the concentration gradient. Because sodium is a positively charged ion, it will change the relative voltage immediately inside the cell relative to immediately outside. The resting potential is the state of the membrane at a voltage of -70 MV, so the sodium cation entering the cell will cause it to become less negative. This is known as depolarization, meaning the membrane potential moves towards zero. The concentration gradient of sodium ion is so strong that it will continue to enter the cell even after the membrane potential has become zero, so that the voltage immediately around the pore begins to become positive. The electrical gradient also plays a role as negative proteins below the membrane attract the sodium ion. The membrane potential will reach positive 30 MV by the time sodium has entered the cell. As the membrane potential reaches plus 30 MV, other voltage gated channels are opening in the membrane. These channels are specific for the potassium ion. A concentration gradient acts on potassium ion as well. As potassium ion starts to leave the cell, taking a positive charge with it, the membrane potential begins to move back towards its resting voltage. This is called repolarization, meaning that the membrane voltage moves back towards the -70 MV value of the resting membrane potential. Repolarization returns the membrane potential to the -70 MV value that indicates the resting potential, but it actually overshoots the value. The potassium ions reach equilibrium when the membrane voltage is below -70 MV, so a period of hyperpolarization occurs while the potassium channels are open. Those potassium channels are slightly delayed in closing, accounting for this short overshoot. What has been described here is the action potential, which is presented at a graph of voltage over time. It is the electrical signal that nervous tissue generates for communication. The change in the membrane voltage from -70 MV to raised to plus 30 MV at the end of depolarization is a 100 MV change. That can also be written as a 0.1 V change. To put that value in perspective, think about a battery. An A battery that we might find in a television remote has a voltage of 1.5 volt or a 9 volt battery. The rectangular battery with two posts on one end is obviously 9 volt. The change seen in the action potential is 1 or 2 orders of magnitude less than the charge in these batteries. In fact, the membrane potential can be described as a battery. A charge is stored across the membrane that can be released under the correct condition. A battery in your remote has stored a charge that is released when you push a button. So now the question is, what initiates the action potential? The description above conveniently glosses over that point, but it is vital to understanding what is happening exactly. The membrane potential will stay at the resting voltage until something changes. The description above just says that a sodium ion channel opens. Now, to say a channel opens does not mean that one individual transmembrane protein changes. Instead, it means that one kind of channel opens. There are few different types of channels that allow sodium to cross the membrane. A ligand-gated sodium channel will open when a neurotransmitter binds to it, and a mechanically gated sodium channel will open when a physical stimulus affects a sensory receptor, like pressure applied to the skin compresses a touch receptor. Whether it is a neurotransmitter binding to its receptor protein or a sensory stimulus activating a sensory receptor cell, some stimulus gets the process started. Sodium starts to enter the cell and the membrane becomes less negative. A third type of channel that is an important part of depolarization in the action potential is the voltage-gated sodium channel. The channels that start depolarizing the membrane because of a stimulus help the cell to depolarize from -70 MV to -55 MV. Once the membrane reaches that voltage, the voltage-gated sodium channel opens. This is what is known as threshold. Any depolarization that does not change the membrane potential to -55 MV or higher will not reach threshold and thus will not result in an action potential. Also, any stimulus that depolarizes the membrane to -55 MV or beyond will cause a large number of channels to open and an action potential will be initiated. Because of the threshold, the action potential can be likened to a digital effect. It either happens or it does not. If the threshold is not reached, then no action potential occurs. If depolarization reaches -55 MV, then the action potential continues and runs all the way to +30 MV, at which potassium causes repolarization, including the hyperpolarization of a shoot. Also, those changes are the same for every action potential, which means that once the threshold is reached, the exact same things happens. A stronger stimulus which might depolarize the membrane will pass threshold, will not make a bigger action potential. Action potentials are all or none. Either the membrane reaches the threshold and everything occurs as described just previous, or the membrane does not reach the threshold and nothing else happens. All action potentials peak at the same voltage, positive 30 MV, so one action potential is not bigger than another. Stronger stimuli will initiate multiple action potentials more quickly, but the individual signals are not bigger. Thus, for example, you will not feel a greater sensation of pain or have a stronger muscle contraction because of the size of the action potential, because they are not defined sizes. As we have seen, the depolarization and repolarization of an action potential are dependent on two types of channels, the voltage-gated sodium ion channels and the voltage-gated potassium channel. The voltage-gated sodium channel actually has two gates. One is the activation gate, which opens when the membrane potential crosses -55 MV. The other gate is the inactivation gate, which closes after a specific period of time on the order of a fraction of a millisecond. When a cell is at rest, the activation gate is closed and the inactivation gate is open. However, when the threshold is reached, the activation gate opens, allowing sodium ion to rush into the cell. Timed with the peak of depolarization, the inactivation gate closed. During repolarization, no more sodium can enter the cell. When the membrane potential passes -55 MV again, the activation gate closes. After that, the inactivation gate reopens, making the channel ready to start the whole process over again. The voltage-gated potassium channel has only one gate, which is sensitive to a membrane voltage of -50 MV. However, it does not open as quickly as the voltage-gated sodium channel does. It might take a fraction of a millisecond for the channel to open once that voltage has been reached. The timing of this coincides exactly with when the sodium ion flow peaks. Two voltage-gated potassium channels open just as the voltage-gated sodium channels are being inactivated. As the membrane potential repolarizes and the voltage passes -50 MV again, the channel closes again with a little delay. Potassium continues to leave the cell for a short while and the membrane potential becomes more negative, resulting in the hyperpolarization overshoot. Then the channel closes again and the membrane can return to the resting potential because of the ongoing activity of the non-gated channels and the sodium-potassium pump. All of this takes place within approximately 2 milliseconds. While an action potential is in progress, another one cannot be initiated. That effect is referred to as the refractory period. There are two phases of the refractory period, the absolute refractory period and the relative refractory period. During the absolute phase, another action potential will not start. This is because of the inactivation gate of the voltage-gated sodium channel. Once that channel is back to its resting conformation less than -55 MV, a new action potential could be started, but only by a stronger stimulus than the one that initiated the current action potential. This is because of the flow of potassium out of the cell. Because that ion is rushing out, any sodium ion that tries to enter will not depolarize the cell but will only keep the cell from hyperpolarization. Now come to the point, the propagation of the action potential. The action potential is initiated at the beginning of the action, at what is called the initial segment. There is a high density of voltage-gated sodium channel so that rapid depolarization can take place here. Going down the length of the action potential is propagated because more voltage-gated sodium channels are opened as the depolarization spreads. This spreading occurs because sodium enters through the channel and moves along the inside of the cell membrane. As the sodium moves or flows a short distance along the cell membrane, its positive charge depolarizes a little more of the cell membrane. As that depolarization spreads, a new voltage-gated sodium channels open and more ions rush into the cell, spreading the depolarization a little further. Because voltage-gated sodium channels are inactivated at the peak of the depolarization, they cannot be opened again for a brief time, the absolute refractory period. Because of this, depolarization spreading back toward previously open channels has no effect. The action potential must propagate toward the action terminals. As a result, the polarity of the neuron is maintained as mentioned above. Propagation, as described before, applied to unmyelinated axons. While myelination is present, the action potential propagates differently. Sodium ions that enter the cell at the initial segment starts to spread along the length of the axon segment, but there are no voltage-gated sodium channels until the first node of Ranvier. Because there is no constant opening of these channels along the axon segment, the depolarization spreads at an optimal speed. The distance between nodes is the optimal distance to keep the membrane still depolarized above threshold at the next node. As sodium spreads along the inside of the membrane of the axon segment, the charge starts to dissipate. If the node were any further down the axon, but the depolarization would have fallen off too much for voltage-gated sodium channels to be activated at the next node of Ranvier. If the nodes were any closer together, the spread of propagation would be slower. Propagation along an unmyelinated axon is referred to as continuous conduction. Along the length of a myelinated axon, it is solitary conduction. Continuous conduction is slow because there are always voltage-gated sodium channels opening and more sodium is rushing into the cell. Solitary condition is faster because the action potential basically jumps from one node to the next, the soldier to live, and the new influx of sodium renews the depolarized membrane. Along with the myelination of the axon, the diameter of the axon can influence the speed of conduction. Much as water runs faster in a wide river than in a narrow creek, sodium ion-based depolarization spreads faster down a wide axon than down a narrow one. This concept is known as resistance and is generally true for electrical wires or plumbing, just as it is true for axons, although the specific conditions are defined at the scales of electrons or ions versus water in a river. Revise it, I am damn sure you will require less time to remember all these structures and processes. Write me at the comment box how much you enjoyed and time take to remember the nervous tissue anatomy after reviewing today's lecture. Next episode, we will discuss on the communication between neurons. If you like this video, please like, share and comment freely. Don't forget to subscribe our channel. If you already subscribed, then thanks a lot. After subscribing our channel, please click on the bell button for getting the regular updates. I am coming with another video very shortly. Till then, goodbye.